This is Unit 16, Blood. In this video, we'll look at Part A, General Principles, Part B, Blood Plasma, and Part C, Red Blood Cells. This unit is divided into two videos. First video, this one, includes Part A, B, and C. The second video includes Part D, E, and F. Under Part A, General Principles of the Circulatory System, there are three parts to the circulatory system. One, the fluids. These are located in compartments, including the blood in the cardiovascular system, lymph in the lymphatic system, and interstitial fluid found among the tissue cells. The fluids can shift between compartments, as you see by the red arrows. The fluids are extracellular fluids, ECF. As you'll see, they're pretty much identical to one another. The intracellular fluid is the cytoplasm of the cell. The second component is the lymphatic system. It includes lymph vessels, lymph nodes, lymph tissue, and the third part is the cardiovascular system. It consists of the heart and blood vessels. Among the blood vessels, arteries transport away from the heart, veins transport to the heart. The cardiovascular system has a special inner lining. It's called endothelium inside the vessels and endocardium inside the heart. The arrows indicate the direction of fluid movement. Notice that fluids move out of the capillaries into the interstitial fluid compartment, or from the interstitial fluids back into the capillaries. By the way, when I use the term circulatory system, I mean the overall systems as we just described. If I refer to the heart and blood vessels, I'll refer to it as the cardiovascular system. The circulatory system has a number of functions. Transport is one. Immunity, defense against disease, is another. Self-protection, clotting and buffering. Part B, blood plasma. Plasma and the formed elements make up the blood. Plasma is the liquid part. The formed elements are the cells. There are three types: red blood cells, abbreviated RBC, white blood cells, WBC, and platelets. The size of each component in the graph represents a rough idea of the quantity of each. The characteristics of plasma. The percentage, for example. In plasma, in males, 60% of the blood is plasma. In females, it's about 64%. The viscosity. Plasma is two times as thick or viscous as water. Whole blood is 5 times as viscous. Whole blood now is blood with the red blood cells. Osmolarity is due mainly to sodium, proteins, and the red blood cells. Composition of plasma. It's 90% water, 10% dissolved substances. These include proteins, most of which are produced in the liver, gases, electrolytes, and molecules. Proteins have many different functions. They provide colloidal osmotic pressure. This is the osmotic pressure of the blood due to the presence of proteins. They're involved in transport, immunity. Many exist as systems in the blood, such as clotting proteins, kinins, and complement. These are involved in immunity. The types of proteins include albumins. They function to transport lipids mainly. 3.8 to 5.4 grams per 100 milliliters of blood is a normal range. Globulins, these function in transport and immunity. They make up roughly 2.2 to 3.4 grams per 100 milliliters of blood. Notice that measurements in terms of 100 milliliters is a convenient way to designate quantities in the blood. The types of globulins include alpha, that functioning in transport, beta, that function in transport, and gamma, that are the antibodies. They make up about 20% of the plasma proteins. The gases include oxygen, carbon dioxide, and nitrogen. The electrolytes, these are ions, include such as sodium, potassium, and chloride ions. In addition to that, there are bicarbonate ions, and the proteins are often ionized. There are molecules in the plasma, amino acids, for example, fatty acids, glucose. The normal range is roughly 70 to 110 milligrams per 100 milliliters of blood. Hormones, including thyroid hormone and growth hormone. Cholesterol, roughly 111 to 279 milligrams per 100 milliliters of blood. Notice that we refer to cholesterol frequently. It's not a harmful substance unless it's found in excess. You have to have cholesterol because it's the precursor for many other substances. Bilirubin is also found in the plasma. There may be none or up to 0.8 milligrams per 100 milliliters of blood. We'll come back to bilirubin shortly. Urea is another type of molecule in the blood. Notice that the distribution of positive and negative ions is almost identical in the extracellular fluids of the body, plasma, lymph, and interstitial fluid. The intracellular fluid, that is the cytoplasm, on the other hand, is different. Notice that we've referred to 100 milliliters. It's the same as 1/10 of a liter or 1 deciliter, abbreviated BL. Part C, red blood cells, including structure, life cycle, and blood types. Red blood cells are also referred to as RBC or erythrocytes. As we mentioned, they're the most common of the formed elements. About roughly 5 million per cubic millimeter is found in the blood. That amounts to 5 billion per cubic centimeter, or 25 trillion per human. When we talk about trillions, that's an inconceivably large number. If you tried to count from 1 to a trillion, one number per second, it would take you thousands of years to count to a trillion. The quantity differs slightly in males and females. Males have roughly 5.1 to 5.8 per cubic millimeter. Females have 4.3 to 5.2 per cubic millimeter. The structure of red blood cells is in the form of a biconcave disc, 7.5 microns in diameter. If you look at a top view of the red blood cell, it looks round. If you turn that down and look at it from side view, it looks flat. And if you slice it in half, then you can see the two concave surfaces. Red blood cells lack the usual cellular organelles. They are essentially sacs filled with hemoglobin. Let's look at the structure of hemoglobin. It contains 4 globin proteins. 2 of them are alpha and 2 are beta globulins. It contains 4 hemes, each composed of bilirubin and iron. 50% of the total body iron is found in the hemoglobin. 25% is found in cytochromes and other heme-containing proteins. We discussed cytochromes in the cellular unit under metabolism. 25% are found in the liver, bound to a protein called ferritin. The figure shows one alpha and one beta protein in the hemoglobin molecule. Remember, there are actually two of each. Hemoglobin concentration, 13 to 18 grams per 100 milliliters in men, 12 to 16 grams per 100 milliliters in women. Why fewer in women? Well, women lose some hemoglobin during the menstrual cycle. The function of red blood cells is to transport oxygen. Hemoglobin plus oxygen forms a molecule called oxyhemoglobin, HbO2. And here's the formula. Hemoglobin plus oxygen gives oxyhemoglobin. Now, actually, since the hemoglobin has 4 hemes, it binds to 4 oxygens, so this little equation is not a balanced equation. Arterial blood, we refer to it as oxygenated blood. It's bright red. It's found in the arteries. They contain 100% oxyhemoglobin. There is one exception to that. The exception is the pulmonary vein, which contains arterial blood. That makes sense. After all, the pulmonary vein comes from the lungs where the blood has picked up oxygen and transports the blood back to the heart. Venous blood is deoxygenated blood. It has a dark red, almost a blue look. Veins, with one exception, contain venous blood. What percentage of hemoglobin would you say is found in venous blood? We said it's deoxygenated. Would it be 0%? No, in fact, it contains 75% oxyhemoglobin. So you see, at rest, the blood has only given up 25% of its hemoglobin as it circulates through the tissues. The exception to veins containing venous blood is the pulmonary artery. The pulmonary artery travels through the lungs to pick up oxygen. Since there are so many red blood cells, they don't last long. Their lifetime is roughly 120 days. And again, because of the great number of red blood cells, every second, 2.5 million have to be destroyed. That's 100 billion a day. Red blood cells wear out, and they tend to break down after a while. A major problem is disposal of the hemoglobin. Not so big a problem disposing of the rest of the red blood cell. Macrophages are involved. These are mainly in the spleen, but also in the liver, lymph system, lungs, and loose connective tissue. Although the spleen is a major organ in red blood cell breakdown, a splenectomy does not seem to affect red blood cell lifespan. Here's a view. Here's the hemoglobin molecule and the macrophage. The fragments are taken into the macrophage. The macrophage hemoglobin is split into globin and heme. Globin is recycled, and the heme is split into iron and bilirubin. The iron is recycled. It's bound to protein to be transported, transferrin, and then most is taken to the bone marrow. Some is taken to the liver for storage in the form of the ferritin, which we mentioned. Bilirubin then is released from the macrophage into the blood. In the blood, the bilirubin combines with albumin to make it water soluble. It's transported to the liver in the plasma, taken into the liver where the bilirubin is bound to glucuronic acid to make it water soluble. The enzymes are required for this step. From the liver, the bilirubin is transported through the bile ducts to the intestines, and in the intestines, The bilirubin is oxidized through several intermediate steps by intestinal bacteria. It produces a brown substance that gives the feces a brown color. Newborns lack the bacteria, so unoxidized bilirubin, yellow in color, gives the bright yellow color to newborn feces. The bilirubin ends up being excreted with the feces. Some intermediates are transported to the kidneys. They're changed to a yellow substance called urobilin and excreted in the urine. If levels of bilirubin increase in the circulation, jaundice results. Now, jaundice is often referred to as yellow jaundice, but that's a redundant term because jaundice means yellow, so we don't call it yellow. The causes of jaundice in newborns includes a deficiency of the enzymes needed to bind bilirubin to the proteins. Premature babies may lack these enzymes. A high hematocrit is the percentage of red blood cells, may lead to jaundice. Delay in clamping of the umbilical cord may be another cause. Decreased feeding of newborns slows bowel motility and allows recirculation of bilirubin in the newborn. Bilirubin causes some problems because it binds to cell membranes. It's lipid soluble. It may cause cell death. This may lead to cerebral palsy, mental retardation, deafness, and seizures. What about the manufacture of red blood cells? Well, we said 2.5 million are destroyed every second. Therefore, 2.5 million must be produced every second. Manufacture of red blood cells is called erythropoiesis. It occurs mainly in the red bone marrow of the vertebrae, ribs, and sternum. It occurs by these steps. Stem cells become a CFUE, that is a colony-forming unit, and the colony-forming unit becomes a proerythroblast, also called hematocytoblast. It makes hemoglobin. Proerythroblast then becomes a reticulocyte. It releases the nucleus and exits the bone marrow. Reticulocytes then mature into red blood cells. This process is controlled by the hormone erythropoietin. It's made in the kidneys and liver by this mechanism. Decrease in oxygen, this is called hypoxia, is sensed by the liver and the kidney. It increases erythropoietin production and release. The red bone marrow, and hence the target, is stimulated, and the action is to increase the red blood cell production. Notice that by keeping in mind the general scheme of hormone function, this helps you remember details of certain processes. Anemia is a decrease in red blood cells or hemoglobin. It includes nutritional anemia, decreased iron in the diet, hemorrhagic anemia due to bleeding, pernicious anemia, decreased vitamin B12, which is necessary for red blood cell manufacture, hemolytic anemia, increased breakdown of red blood cells. This includes sickle cell anemia, although some textbooks list sickle cell as a separate category. Hypoplastic anemia, decreased red blood cell production. Aplastic anemia, no red blood cell production. Blood type depends on proteins on the red blood cells and involves proteins in the plasma. Red blood cells contain proteins called antigens or agglutinogens. They're in the plasma membrane. They include one called antigen A. We use a capital A to designate that. As we've said already, it's determined by a gene, capital I subscript capital A. There's another one called antigen B. We'll use uppercase B for that. It's determined by the gene IB. The absence of A and B is determined by gene I. There's another one called the antigen RH. Actually, this is 6 or more antigens, but one is most important, and for convenience, we'll assume just one exists. There are many other antigens also present. Blood plasma contains proteins called antibodies, or agglutinins. They're shaped roughly like a Y with 2 antigen binding sites. There's one called anti-A, designated with the lowercase a, another one anti-B, designated with lowercase b, and then there's one called anti-RH, and there are others. Many of the antigens and antibodies are complementary to one another. This is a critical point. Antigens can be abbreviated AB. Antibodies will abbreviate AB. Complementary antigens and antibodies will bind to one another because their shapes match. This antibody, for example, might bind to this antigen. Examples of complementary antigens and antibodies are capital A antigen and little a antibody, B and B, RH and anti-RH. Normally, a person's blood will not contain complementary antigens and antibodies. If complementary antigens and antibodies are present, they will bind, causing the red blood cells to stick together, that is, to agglutinate. Note, agglutination is not clotting, it's sticking together. Blood types are based on the antigen on the red blood cell. For example, one group of blood types is the ABO group. There's blood type A, for example. It contains antigen A on its red blood cells and anti-B, or little b, in the plasma. Blood type B. Blood contains B antigen on the red blood cells and anti-A antigen in the plasma. Blood type AB. It contains both antigens A and B, but it contains neither antibody in the plasma. And then there's blood type O. Blood type O has neither antigen A nor antigen B, but it has both anti-A and anti-B antibodies in the plasma. The RH group, that's another family. RH positive people have RH antigen present on the red blood cells. That person will never have anti-RH antibodies in the plasma. RH negative people, they never have the anti-RH antigen on the red blood cells, but sometimes they have the anti-RH antibodies in the plasma. You'll understand what we mean as we progress on. Let's look at transfusions as they affect the ABO group. In the following figures, the antigen on the red blood cells is shown at the top, the antibody in the plasma at the bottom, and the blood type beneath the figure. A transfusion involves a donor and a recipient. Generally, the danger is due to the donor's red blood cells agglutinating as they enter the recipient. If the antigens on the donor's red blood cells are complementary to the antibodies in the recipient's plasma, the two will bind. Less risk is involved if the antigens on the recipient's red blood cells are complementary to the donor's antibodies. There's less agglutination due to dilution of the donor's antibodies and various other factors. The transfusion, as we said, involves a donor and a recipient. Remember, the danger is due to the donor's red blood cells agglutinating as they enter the recipient. Here are 4 examples of transfusions from a donor to a recipient. Each oval is a person. As we mentioned already, the antigen is shown at the top, the antibody at the bottom within the figure, and the blood type is indicated below each person. On the next slide, you'll be asked to complete the missing information. You'll also determine which transfusions are safe. Antigens, again, we use the symbol Ag, and for antibodies, we use the symbol Ab. Person number 1 is the donor. This person has blood type A, therefore this person has antigen A and antibody B. Person number 2 has antigens A and B, therefore this person must have no antibodies and has blood type AB. Is this transfusion safe? Of course it is. You can't have a reaction between something and nothing. Person number 3 is a donor. This person has antibody A in the plasma. So what antigens and blood type does this person have? Antigen B, therefore blood type B. Person number 4 is the recipient. This person has blood type B, so this person has antigen B and antibody A. Is this a safe transfusion? Yes, it is, because there are no complementary antigens and antibodies present. Person number 5 has antigen A in the plasma on the red blood cells. So which antibody and blood type apply to this person? Antibody B, lowercase b, and blood type A. Person number 6 is the recipient. This person has blood type O, therefore what antigens are present and what antibodies are present? No antigens, but antibodies A and B. What about this transfusion? Is it safe? No, it is not, because the A antigens on the incoming red blood cells will react with the complementary A antibodies in the recipient, causing agglutination of the blood. Person number 7 has blood type AB, therefore has A and B antigens and no antibodies. Person number 8 is the recipient. This person has no antibodies, but he or she does have the antigens A and B, therefore the blood type is AB. Is this a safe reaction? Yes, it is. Again, you can't have a reaction between something and nothing. Person with which blood type can accept blood from an O donor? Any, because a person with blood type O has no antigens that will react with whatever antibodies the recipient has. Therefore, a person with blood type O is a universal donor. A person with which blood type can give blood to an AB recipient? Well, the AB recipient has no antibodies. Therefore, whatever antigen is present on the donor's red blood cells will find nothing to react to. So an AB person can accept from any donor, and a person, therefore, with type AB blood is a universal recipient. Let's look at the RH group. Now remember, we said there are more than 6 RH types. Let's assume there's only one, because one of them is more important, and it allows us to understand it a little better. Let's consider an Rh positive donor and an Rh negative recipient, and assume the 2 individuals have never been exposed to someone else's blood. Indicate the antigens and the antibodies in both. The Rh positive person, the antigen is present. After all, that's what determines the blood type. The anti-Rh antibody, however, is absent. In the Rh negative person, the Rh antigen is absent, and the anti-Rh antibodies are absent. Would this be a safe transfusion? Well, it would, because you can't have a reaction between something and nothing. But there's a very important stipulation, as we'll address in a moment. After the transfusion, the immune system of the recipient starts to make anti-Rh antibodies in response to the Rh antigens. Assume a second transfusion is required from the Rh positive donor. Is this transfusion safe? No, because the Rh antigen in the donor's blood will react with the anti-Rh antibodies in the recipient's blood. So when I say sometimes, it means that an Rh negative person will have anti-Rh antibodies only after exposure to the Rh positive blood. There's a disease called Erythroblastosis fetalis. This is the Rh disease of newborns. In the first pregnancy, if the mother is Rh negative and the fetus is Rh positive, which antigens and antibodies are present in the mother and the fetus? Well, if the mother is negative, she would lack the Rh antigen. If she has never had a transfusion or been exposed to positive blood, she would also lack the anti-Rh antibody. The fetus, being Rh positive, would have the Rh antigens, but would also lack the anti-Rh antibodies. So the mother has none of each. The fetus has antigens, but no antibodies. During birth, some of the blood is exchanged between mother and fetus. Also, some of the antibodies may cross the placenta because these are small antibodies. The mother's immune system will develop anti-Rh antibodies, not many after the first pregnancy. Is this a problem with either the mother or the fetus at this time? No, no problem at this time because it takes several weeks for the mother to develop the antibodies. What about the second pregnancy? The next time the mother becomes pregnant, some maternal antibodies will cross the placenta, the mother to fetus. Since the fetus is Rh positive, some red blood cell agglutination and destruction in the fetus will occur. One problem is the red blood cells break down and release hemoglobin, which forms bilirubin, leading to jaundice. Also, more fetal red blood cells leak into the mother's circulation at birth, and the mother's immune system now develops more of the anti-Rh antibodies. Later pregnancies, if the fetus is Rh positive, more maternal antibodies will cross into the fetus, causing a more severe reaction, more agglutination of the fetal red blood cells. This is prevented with an injection of anti-Rh antibodies that prevents the mother from developing her own anti-Rh antibodies. The exact mechanism is not entirely understood. And this is the end of parts A, B, and C of Unit 16. You're now ready to go to Part D, E, and F in Unit 16.